We now know the basics about prokaryotic cells, but cells get much more complicated than this. Human beings and all other animals are made of eukaryotic cells, which contain a number of components called organelles that are not present in prokaryotic cells, some of which resemble individual organisms unto themselves. Because they are more complex, we know that eukaryotic cells evolved from prokaryotic cells. Endosymbiotic theory proposes that billions of years ago, there were many different types of bacteria floating around, all of which were unicellular. Some of these species of bacteria acquired unique functions, like the ability to perform photosynthesis or breathe oxygen. It is believed that larger host cells then enveloped some of these smaller bacteria, and instead of digesting them, a mutual dependency arose, each relying on the other for some crucial function. The smaller cell received protection, while the larger one reaped the benefits of some kind of energy-producing process. This is where the name endosymbiotic comes from. Endo means inside, and symbiosis refers to organisms living and working together. These smaller bacteria grew more specialized over millions of years of evolution, and eventually became some of the organelles we find within a single eukaryotic cell. So what are these organelles exactly? To answer that, let's first make the distinction between plant cells and animal cells. These are both examples of eukaryotic cells, but they differ in the precise components within. Let's first take a look at animal cells, which are the kind inside you and me, since humans are considered part of the animal kingdom. The first thing we want to point out is that while prokaryotic cells have their genetic information just floating in the middle of the cell, eukaryotic cells store their genetic information inside a nucleus. This nucleus has its own membrane, which, unlike the cell membrane, is actually a double membrane. And this nuclear envelope only lets certain things in or out by passing through tiny holes called pores. In this way, it keeps all the genetic material, or chromatin, separate from the cytoplasm of the cell. Within the nucleus is a smaller area called the nucleolus, where ribosomal RNA is synthesized, as well as the parts of the ribosomes themselves. The large and small subunits of the ribosome are formed, which then exit the nucleus and assemble in the cytoplasm. Some of these ribosomes can be found floating around in the cytosol, but some are stuck to the surface of the nucleus or other organelles, like barnacles on a boat. Ribosomes, which are present in both prokaryotic and eukaryotic cells, are made of ribosomal RNA and proteins, and these in turn synthesize all the proteins the cell needs to perform its various functions. Other components of the cell come together to make up the endomembrane system. This is a set of membrane-bound regions, which consists of the nuclear envelope, the rough and smooth endoplasmic reticulum, Golgi apparatus, and lysosomes. The rough endoplasmic reticulum is a network of membranes that enclose the ER lumen, which is the stuff inside, consisting of sacs called cisternae. It's called the rough endoplasmic reticulum because its surface is studded with ribosomes, giving it a rough appearance. These ribosomes synthesize polypeptides that are inserted directly into the ER lumen, where they are typically folded and modified by enzymes in the ER membrane, and sometimes affixed with a carbohydrate to make glycoproteins. These are often intended for secretion from the cell, so they gather into a vesicle, which is like a bubble that buds from the ER membrane and then makes its way to the cell membrane, thus releasing its contents to the extracellular space. The rough ER also synthesizes phospholipids that can be delivered to other parts of the endomembrane system, and it takes proteins synthesized by the ribosomes that will be cell membrane proteins, embeds them in its own membrane, and then delivers them via similar transport vesicles. The rough ER is connected to the smooth ER, which is different in that it is not studded with ribosomes. It also has a wider variety of functions, like synthesis, metabolism, and storage of calcium ions to be used for signaling. The smooth ER produces phospholipids and steroids, like sex hormones. Enzymes in the smooth ER can detoxify drugs and poisons by adding hydroxyl groups to enhance water solubility and flush them from the body. Next in the endomembrane system is the Golgi apparatus. Most transport vesicles, like the ones going from the ER to the cell membrane, stop off here first. The things inside the vesicles, like proteins, are modified and stored, and later sent to where they need to go. The Golgi apparatus consists of a series of stacks, and there is a cis face, usually facing the ER, where vesicles arrive, and a trans face, where vesicles leave. In between, molecules will be modified with sugars removed or substituted, and generally prepared for their eventual function. This is all done piecewise as the components move from one cisterna to the next, as each of these sections has its own set of enzymes. Before sending things off, products are given molecular tags like phosphate groups, which is kind of like an address on a piece of mail to target things for various parts of the cell. And some vesicles also have certain molecules on their surfaces that can be recognized by specific organelles, which helps target vesicle delivery. Another organelle is the lysosome. This is a sac containing an environment that is much more acidic than the rest of the cell, and it holds enzymes that digest large molecules via hydrolysis, which is the breakdown of a compound due to reaction with a water molecule. When food enters a cell through a vesicle by a process called phagocytosis, multiple vesicles fuse to form a vacuole. Then a lysosome will fuse with the vacuole and break apart the big chunks of food into smaller bits. Lysosomes can also digest damaged organelles that need to be dismantled in a process called autophagy. This allows the cell to maintain optimal functionality. There are a few organelles that are not part of the endomembrane system, and a crucial one is the mitochondrion. There are hundreds of mitochondria in most eukaryotic cells, and these are the sites that carry out cellular respiration, which generates most of the energy the cell needs to go about its business. Cellular respiration occurs in 3 steps: glycolysis, the citric acid cycle, and oxidative phosphorylation. Beyond this, mitochondria also have 2 membranes. The outer membrane is smooth, but the inner membrane makes many folds called cristae. The area in between the 2 membranes is the intermembrane space, while the innermost part is called the mitochondrial matrix. The matrix contains circular DNA molecules called mitochondrial DNA and ribosomes, as well as many of the enzymes that carry out cellular respiration, while some of these enzymes can be found in the inner membrane itself. Another organelle that also has metabolic function is the peroxisome. This contains enzymes that perform oxidative processes, which make hydrogen peroxide as a byproduct. These work with mitochondria by breaking down fatty acids into smaller components that are ready for cellular respiration. That's all the organelles, so what else is in there? Well, the cellular components are typically tethered to something called the cytoskeleton. This is a network of fibers that stretch throughout the cytoplasm of the cell and keep everything nice and organized. These fibers consist of hollow microtubules made of tubulin dimers, microfilaments made of two intertwined strands of actin, and intermediate filaments made of coiled keratins. Beyond organization, these also determine and maintain the shape and structural integrity of the cell. Sometimes vesicles utilize motor proteins to deliver their contents to their destinations by moving along microtubules, like from the ER to the Golgi apparatus, or from the Golgi apparatus to the cell membrane. This is a process that requires ATP to power, and two components move sequentially along the microtubule in an action that looks a lot like walking. In animal cells, microtubules grow outwards from a centrosome, which is located near the nucleus and consists of a pair of centrioles, which have microtubules arranged in a ring, 9 sets of 3 to be exact. Now that we know a bit about the structure and function of each organelle, we can almost view the cell as a tiny factory, with each component serving a specific purpose. The plasma membrane is like the security guard at the gate, deciding what can go in or out. Once inside, we see the cytoskeleton, which is the system of girders that provides structure, and the cytoplasm, which is the factory floor. The nucleus is the central office where the boss sits, giving out orders from the genetic instructions. The endoplasmic reticulum is the assembly line, and the ribosomes are the little factory workers, building all the proteins the cell needs. The Golgi apparatus is the sorting and shipping center. The lysosomes are the maintenance and disposal workers, while the mitochondria are the furnace or power plant that powers the whole show. Of course, this is just an analogy. None of these organelles have any sentience, but it illustrates how a cell is much greater than the sum of its parts, a living system with components that cooperate for survival. In addition, after learning a bit about organelle structure and function, endosymbiotic theory should become quite compelling. It is now clear that membrane-enclosed organelles like the nucleus, endoplasmic reticulum, and Golgi apparatus probably originated from deep folds in the plasma membrane, whereas mitochondria, as well as chloroplasts, which we will get to next, must have begun as completely separate organisms, since they have their own DNA and membranes that are different from the cell membrane. All of these diverse structures eventually came to serve as compartments for different cellular activities, which over an immense time span resulted in things as complex as the plant and animal cells on Earth today. We still have more to learn about cells, and we are just scratching the surface of their complexity. But we've now gone from biomolecules to prokaryotic cells to eukaryotic cells. So let's learn about another type of eukaryotic cell next.